As you can see from the coagulation analysis here, the serum base is working effectively. This critical diagnostic information.
The serum-based results are conclusive. We can proceed with the DA18 protocol immediately. The accuracy is unparalleled. This facility represents the intersection of high-risk biological handling and the advanced in industrial. Module D-A18, origin confirmed.
Manifest matches. Proceed to rearing zone.
The sample is instantly routed via sterile capillary tube for immediate spectroscopic analysis, providing preliminary data on the protein markers.
The physical and operational separation is critical. On the left, raw Dispholidus typus enters the extraction suite.
The material divergence is showcased here. Process A prepares the venom, contrasting with process B, where
enzyme load is complete.
Initiating digestion sequence B18. Confirming temperature at 37 Celsius.
The crude venom sample is immediately placed into a cryogenic flash freezer, instantly
The concentrated hemotoxin enters the final, high-resolution fractionation system, a specialized
The UV sterilization levels on batch 4 optimize the decontamination process.
Agreed, the throughput is exceeding expectations.
The successful initial separation yields a clearer supernatant, which then undergoes diafiltration.
The specialized serum, Hemotoxic Research Venom Model D-A18, is loaded and confirmed. The
The specialized serum base, derived from the Boomslang byproduct, functions as a high-quality diagnostic reagent.
The stabilized B feedstock is now being transferred into the primary filtration manifold, marking the official start of specialized serum-based manufacturing.
The B feedstock undergoes a primary clarification step using a large disc stack centrifuge designed for
The highly concentrated protein solution is routed through a sterile heat exchanger for pasteurization. This ensures The filled dropper bottles immediately move to the capping station, where color-coded caps are automatically sorted and affixed.
Ensure the DA18 kits are secured in the cryo modules.
Already done. We are ready for dispatch.
Temperature is stable at 4 degrees Celsius.
Scan complete. The cold chain is verified.
The specialized serum base is placed next to the sample wells, ready for use. The professional then verifies
correlation between the patient samples
and these clotting times is strong. Absolutely. This confirms the specialized serum.
This image signifies the completion of the product's life cycle and its successful application in a clinical setting.
The diagnostic kits are all accounted for. Please sign on line 3.
Thank you. The cold chain looks stable.